Recording today is, of course, 9/10/18. So we said that all populations have variation. There's our bacterial wallpaper. That antibiotic resistance happens through natural selection. And we've talked about the first 2 points of natural selection. And now we're going to talk about that third. Individuals tend to have more offspring than will survive. So how many babies does a bacteria want to grow up to have? All of them. As long as conditions are right, and that gets back to our homeostasis point. Remember we talked about if you're out working with tools in the cold, your hands don't grip things right. Or me, before I've had a cup of coffee in the morning, I can't grip a pencil. As long as the conditions are right, and this is mostly, what are the 2 conditions that really affect bacteria?
Temperature and water.
Temperature and water balance. As long as their temperature range is good, they're going to keep reproducing. Why do we put food in the fridge?
Keep it cool.
Why do we care if it's cold? What do you mean fresh?
Not gonna hurt you.
Preserve it? Why? Well, they'll go there, but they won't reproduce.
They'll
not reproduce. Bacteria require certain temperatures to reproduce. Hence, oh, hey, I even put it in the notes, a fever. Hence, refrigerating your food. Milk. Bacterial populations can grow exponentially. Who knows what exponential means? If I draw you a graph that looks like this, that's an exponential graph. An exponential function, yeah. Y equals x squared. So bacterial populations can actually grow at that kind of rate. That is insane. Now, that is as long as conditions are appropriate for them. When you make life harder for them, that slows them down. But do you think that it's necessary for all of the bacteria that we've produced by here to survive to make something really sick? Probably not. And it's perfectly all right that some of them won't make it. This does relate to bacteria. This used to relate to humans more so than it does now in the industrialized world, you realize? Is anybody here from a family of 12 or more children? I didn't think so. You don't get a whole lot of those. Anybody have a parent who's from a family of 12 or more? Okay, 1. Anybody have a grandparent who was from a family of 12 or more? Anybody know if their great-grandparents were from a family of 12 or more? Okay, and very often we don't have knowledge that far back. Used to be pretty common. My grandpa was one of 10. His father was one of 17 who were born, 11 of whom survived to adulthood. That means his grandparents buried 6 of their children, some of them before the age of 1. That used to be pretty common. You know that? Did you know that?
No.
You didn't understand that? You know what changed one of those big factors for childhood mortality is vaccinations. Kids used to die of measles and mumps and diphtheria and pertussis. Chickenpox doesn't kill as often, but it can be deadly. It's less lethal than some of those others. Diphtheria is pretty lethal. So go to the old cemetery up at the corner of Sprucevale and Eckodale. What's that? Clarkson Presbyterian and Gesundheit. There's a cemetery there with graves dating back to the 1700s. Go look at how many of them are children's graves. Because there are a lot of them. And in some cases you'll see a whole family where you've got 2 or 3, infants, 1-year-olds, 2-year-olds, 5-year-olds who died of what are now preventable childhood illnesses. In most species, individuals tend to produce more offspring than are going to make it. This is so simple, it's ridiculous. You love simple?
Writing.
Oh, writing. Was that sarcasm? Okay. They replicate. What does it mean to replicate? to copy. You're going to replicate the words on this slide in your notes. They replicate their DNA, they copy it, and then identical copies of that DNA move to opposite ends of the cell. Why is there an asterisk by identical?
Because they're identical.
Because
Well, they should be identical. Are they always perfect copies? Are there ever mistakes made?
Yes.
Yes. That's why there's that little asterisk, because they should be identical, but they aren't always. Identical copies of the DNA move to opposite ends of the cell. They divide their cytoplasm. Remember, that's the water jelly. Ooh, sounds delicious. And each bacterial cell gets half the cytoplasm, one copy of the DNA. Easy peasy. You now have 2 new bacteria. They actually call this the mother cell, and they call these the daughter cells. And we can't keep track. We don't know who's got the original DNA. Well, actually, they each have half of the original DNA. We'll talk more about that when we talk about when we talk about DNA and protein synthesis. But it's pretty easy. But it might be slightly more complicated. But the first step in this process, and you're probably going to want to draw this, is certainly to copy that DNA. Copy. Now this is where it gets really tricky in the confines of this. So I now have 2 copies of that DNA floating around in the cell for a moment. It's kind of crowded in there. And this is going to sort of start to grow a little bit bigger. And of course, I messed up my flagella, which is going to allow, let's see if I got that all in one shot. Hey, I did. That's amazing. My 2 strands of DNA to have a little bit of breathing room. And what's going to happen next is, well, now, of course, to do that, do you remember what this is made of? Protein. This is made of fat. It's made of a lipid. So the cell had to produce some extras of those to expand that, didn't it? So it did have to make some more protein to make more cell wall. It had to expand the cell membrane. And then slowly what you're going to start to see is that this is going to kind of, well, that's terrible. This is going to slowly start to pinch in the middle. I'm going to just copy these 2 DNA. Copy, paste. And these little ribosomes, it's kind of like split custody. One new cell gets some of them. One new cell gets the other. And of course, each of these cells had to grow their own flagella. One had the old flagella. I'm going to shrink all of this a little bit. And as long as everything has gone perfectly, we should see
duplication.
We should see exact duplication, yeah. And I always want there to be, though there never is in any of the videos I've seen, a little when they split apart. So I like to imagine it. Well, you can actually. So suddenly what we've got are these two daughter cells, each with their own ribosomes. Now they're going to have to build some new ribosomes because neither of them really get enough ribosomes to make it through. Looks like a pair of very weird eyes, doesn't it?
Sunglasses.
It does. Actually, those would be great sunglasses. You could make that and market it for biology geeks. And pretty soon these can completely move apart from one another. Oh, of course I didn't. Darn it. Well, that was stupid. Okay. That's the process. It really couldn't be a whole lot easier. It's not a complicated process. But it gets you, the last point here is this. Individuals with mutations or features that have some advantage tend to pass on more of their genes. So I'll read this and then shut up while you copy. What might be those individuals with an advantage? Well, if you got a slightly thicker cell wall, you have a slightly different chemical makeup of your cell wall, maybe your proteins are a little bit different. Bacteria who are unharmed by a chemical in their environment, such as an antibiotic. If the better bacteria is the only one left, who gets to reproduce?
No one.
Whose offspring get to make up the population? And we're actually going to do this on Doceri. So as the weak bacteria are killed off and the stronger bacteria, the more resistant bacteria survive, the whole population gets stronger. That's distressing. Yeah, that's not good at all. So how do we prevent? Well, here, let's actually take a look. This was an experiment done, I believe it was at MIT.